Respected teachers, beloved younger and brother and sisters, and my classmates. Today we are all here for this seminar, and I am really grateful to our HOD, Mr. Yudhishthira Goyal sir, for giving us opportunity and chances to appear in this seminar. So my name is Paritri Nayak from Department of Botany, and today my seminar topic is transpiration. So the first thing that comes into our mind is what is transpiration. So the process by which water is released in the form of vapor from the aerial parts of plants are called as transpiration. So there are following types of transpiration that I will mention in this following video. First one is stomatal transpiration, second one is cuticular transpiration, and third one is lenticular transpiration. So the first one, that is stomatal transpiration. The transpiration that occurs in stomata are called as stomatal transpiration. This kind of transpiration occurs during daytime because stomata open during the presence of sunlight and get closed in darkness, and about 80 to 95% of water is released in the form of vapor in this process, and it is the most prominent and accepted one. Then the second one, that is the cuticular transpiration. So the transpiration that occurs in cuticle or cracks or thin layer of cuticle of leaves and stems are called as cuticular transpiration. And in this process, about 5 to 10% of water is released in the form of vapor. And it is a day to night process. We can see that it is a leaf structure, and the upper part above the epidermis is a cuticle. It is a waxy cuticle. And the both of the surface, that is upper epidermis and lower epidermis. In the upper epidermis, cuticle is also present, and below the lower epidermis, cuticle is also present. Then the third one, that is lenticular transpiration. Sometimes transpiration occurs in lenticels. That is the small opening, opening of pulpy tissue surrounding the stems and twigs. And this type of transpiration is called as lenticular transpiration. And in this type, only 0.1% of water is released in the form of vapor. So it's all about the types of transpiration. And among all these, we have learned that 80 to 95% of transpiration occurs in stomatal transpiration. So now I will broadly explain what is the structure of stomata and how the stomata open and get closed, and how transpiration occurs in stomata. So first thing is structure of stomata. Stomata are a microscopic apparatus that present in the epidermis of leaves, green fruits, and herbaceous plants. Stomata never present in roots, and stomata is a biconvex elliptical structure. There are two kidney-shaped epidermal cells, are called as guard cells, that surrounds the stomatal pore. So the cellulose guard cells that surrounds the stomatal pore is thick and inelastic due to the presence of secondary cellulose, or due to the formation of secondary cellulose, and the rest of the cellule is thin and elastic. There are also some epidermal cells that surrounds the guard cells, are called as subsidiary or accessory cells. In case of dicot leaves, stomata are arranged in a scattered manner, but in monocot leaves, stomata are arranged in parallel manner. So the stomata can be found on both the surface, but they are the most prominent and largely found on the lower surface than the upper surface. It's all about the structure of stomata, and now I will explain the same size and number of stomata. So in monocot plants, stomata are less longer cells, but in dicot plants, stomata are ranging of different. Then about the size of stomata. The stomata size vary from species to species, and it can be 3 to 12 micrometer. And the number of stomata also vary from species to species, and it can be thousands to lakhs per unit surface of leaf. Then mechanism of opening and closing of stomata. As I have mentioned earlier, the stomata open during daytime in the presence of sunlight and get closed in darkness. So these are the structure of it is an open stomata and it is a closed stomata. As you can see that in open stomata the guard cells are open, and in closed stomata the guard cells are closed. So this is about the opening and closing of stomata. About describing the opening and closing of stomata, there is a theory which is most widely accepted, that is the Marlet hypothesis or K plus ion theory, and it was given by Fujin and Levitt. So during daytime, that is in the to form oxaloacetic acid, and later oxaloacetic acid is converted into malic acid. After that, malic acid get dissociated and form malate and H plus ion. As the H plus ion concentration increases in guard cell, they remove from the guard cell and in exchange of K plus ion enter to the guard cell. And this process occurs through an H plus K plus anti-transporter, which is an energy dependent process. After that, the H plus and after that the K plus and malic acid are combined to form malate ion. And potassium malate. Then potassium malate enter into the vacuole cell. And as the potassium malate enter into the vacuole cell, the osmotic potential of vacuole increases and endoosmosis of water occurs. As endoosmosis of water occurs, the guard cell becomes turgid and stomata gets open. And transpiration also start or occur. But in night time it is reversed. That is, during night time or in the absence of light, the pH of the guard cell decreases. That is about 5 or 6. And as the pH decreases, the H plus ion and malate ion are combined to form malic acid, and they inhibit. And as a result, the K plus ion releases out from the guard cells to the subsidiary cells. And at that time, abscisic acid is also activated and inhibit the uptake of K plus ion into the guard cells. As the K plus ion releases from the guard cell, the exosmosis of water occurs. That is, water is released out from the guard cell and the guard cell becomes passive. And the result, transpiration stops. This is a diagram of a potassium malate theory. In this process, we can see that here is starch. As the pH increases, the starch is converted into phosphoenol pyruvate with the presence of enzyme peptide. Then phosphoenol pyruvate combined with carbon dioxide and form carbon dioxide to form oxaloacetic acid and then potassium malate. In this process, it is not that elaborately described, but we can see that the 2H plus ion is released and malate is entered into the vacuole cell. And we can see that it is a H plus and K plus anti-transporter. In this process, H plus is released out and K plus is entered with the presence of ATP. And here in vacuole, the malate and potassium are combined and form potassium malate. And at that time, the osmotic potential of the vacuole increases and endosmosis occurs. As a result, guard cell turgid and transpiration occurs. Stomata open and transpiration occurs. This is all about the hypothesis and theory that explain the opening and closing of stomata and how does transpiration occurs in the stomata. Then there are some factors that affect the transpiration process, the rate of transpiration. So there are two categories, that is internal factor and external factor. In internal factor there is stomata, lip area, cuticle and mesophyll. The first one is stomata, which I have mentioned earlier, and major I mentioned earlier that transpiration occurs through stomata. If the presence of stomata increases, the transpiration rate is also increases, and if the presence of stomata decreases, the transpiration rate is also decreases. Then leaf area. There are some plants which have larger leaf area, and there are also some plants which have smaller leaf area. It can observe that it was observed that the plants which have larger leaf area tend to show higher transpiration rate as compared to plants with a lower leaf surface area. Then cuticle.